afternoon, everyone, and welcome to the Botany Bistro. I'm very excited about our topic today and our special guest, so thank you all for joining us. If it's your first time here on the Bistro, welcome. If you're returning, welcome back. We are coming to you live from the Civic Garden Center, which is located in the Avondale neighborhood of Cincinnati, very close to the Cincinnati Zoo, where Megan is working currently. And so it wasn't long for her to come on over here, and we're really happy that she's sharing her time. So just let me give you a little bit of an intro about what the Bistro is. We have been running Bistro sessions every other week for almost a whole year. I'm so excited to say that this is our 24th session together, and we have been talking about basic botany, plant morphology, plant anatomy, plant physiology. We talked about ethnobotany, paleobotany, and recently biotechnology. So this is part 2 of our biotechnology focus. And I started this because I felt like my foundational knowledge in botany has helped me so much in so many aspects of my life, career, and I just wanted to be able to share some of that knowledge with you so that you can build upon it as well. And also, we need more people to be interested in this field because we both know probably that there aren't a lot of college programs, there aren't a lot of career entrance programs that are focused on getting people to do plant science. And we want to see more creativity and more innovation and more ideas. And that's only going to happen with more voices. So hopefully this wets your whistle a little bit on the Bistro today, and then you can go further in your own explorations. And so the Civic Garden Center has been around since 1942. My name is Mary Dudley, and I'm the Ecology Education Manager. I have worked here on and off for 15 years, and it just gets more and more beautiful every day. If you're in the Avondale neighborhood, please stop by and see us. It is so gorgeous right now. And on May 4th, we're going to be having our Plant Grow Fest. So we're going to be having some plants for sale, but also demonstrations and talks focused on urban agriculture and how you can incorporate those best practices into your own lives. All right, so our lesson for today is going to be talking about biotechnology with a real biotechnician right over here. I did give an intro 2 weeks ago. If you want to catch up, that's all found on our archive. So you can either find it on YouTube or you can find it on our website. So you can definitely catch up and see where we were at. We will revisit a couple of those topics that we mentioned before, but I always have something for lunch that pertains to our topic. And so today we are going to be snacking on some strawberries. They're in season, they're delicious, and if you tuned in with me a couple weeks ago, you'll remember that we talked about using strawberries for an experiment to actually extract DNA. And I don't know if any of you tried that experiment. If you did, please let us know. Megan's shaking her head. Yes, strawberry DNA extraction is kind of common in the field. I also have this beautiful strawberry plant with us that we have been growing to show the students that are coming for their field trips about propagation and different techniques for growing hydroponically, in soil, all those different things that you need to know to be a creative gardener. So I just wanted to show off these beautiful strawberries, and we'll snack on those today too. So I hope you're all having a wonderful, healthy lunch here on the Bistro, but let's get into some bite-sized botany. We are going to look ahead for our next session, which will be May 3rd, and we're going to start our ecology series. So we'll have part 1 on May 3rd, and then we will have part 2 later in the month. Now, don't forget, we always have homework, and so your homework for this session is going to be to tell us about your favorite rare or endangered plant. Please share that with us on social media, and be sure to tag us at Civic Garden. I will share that I think my favorite rare plant currently are lady slipper orchids, and they can be found around our state, but they're typically not publicized where they are growing, so you kind of have to know the know. And so if you are familiar with lady slipper orchids and you see them in the wild, look, don't touch, and just enjoy that kind of rare beauty that we have. What's your favorite rare plant?
It's like picking my favorite child, but I think that right now I would say my favorite is the Kentucky clover.
Ooh,
It's like a newly described as science species of clover that's only found in Kentucky. It looks just like a white clover. It looks like the running buffalo clover, but it's very rare, and it's so cool that there's like a new clover in Kentucky that's rare. It's just been recently found, or at least described as its own species. So, yeah, it's a new one.
I love that. So let me introduce our guest, and then we're going to get right into it. So this is Megan Philpott. She's a conservation scientist at the Cincinnati Zoo and Botanical Gardens Center for Conservation and Research of Endangered Wildlife. Those of you that are local might recognize that as the acronym CREW. And so if you go to Cincinnati Zoo, you actually can find her lab and you can walk around and see some of the neat things that they have. So it's over by
the new picnic shelters.
Okay.
By where they're building the new elephant exhibit, depending on how you're familiar with the zoo.
Okay. It's a really cool spot. They have a really neat plant garden over there too. And so the Plant Research Division maintains a cryo-biobank of cryopreserved exceptional plant accessions and works to develop tissue culture and cryopreservation protocols for rare plant species. The lab also maintains the Exceptional Plant Conservation Network as a resource for practitioners working with exceptional species. Megan's work involves the development of shoot-tip cryopreservation protocols for rare species, population genetic analysis of rare plant species, and issues of genetic integrity in exceptional plant conservation. Those are some mouthfuls, so I'm excited to hear you talk about it. She's also involved in building capacity for exceptional plant conservation by developing online resources. She holds a PhD in biology from the University of Cincinnati. Welcome, Megan.
Thanks. I'm excited to be here.
It's awesome. And if you can't tell, she's wearing pawpaw earrings, so you know we're like already best friends. But tell us a little bit about what all that means.
Yeah, so we work on basically seed banking. So seed banking is one of the oldest forms of conservation that has existed. People have been seed banking for generations, hundreds, thousands of years. And that works great for a lot of species, but for a certain number of species that we call exceptional species, they can't be seed banked using those conventional seed banking methods. So sometimes they have seeds, a lot of tropical or desert plants produce seeds that are really sensitive to the drying process or the freezing process, conventional freezing process of conventional seed banking, and so they just don't survive. A lot of plants just don't produce seeds for whatever reason, either naturally or because maybe they've lost a pollinator. They just can't produce seeds anymore. And then the scariest one to me is the seeds that seem like they can be conventionally seed banked and they survive for 1 year, 5 years, 10 years, but then they start to decline and die. And so we call those short-lived seeds. So for all those different species, the best alternative method of long-term preservation for them is cryopreservation, which is the storage of seeds or tissues in liquid nitrogen, which is minus 320 degrees Fahrenheit. It's very, very cold. And so that's kind of what we're working to do. We're targeting these plants that can't be seed banked. Seed banking is much easier and faster than cryopreservation. But for the plants for which that's not an option, we kind of target those and try to get them preserved so that they are available in the future for whatever may happen to the planet or their populations or whatever. So that's kind of our research in a nutshell.
That's awesome. How do you find those partners to work with? Like you said, tropical species and some desert species. So I'm guessing they're not like in our backyard.
They're actually all over. There are some that are in our backyard. Most of our partners will contact us. So there are really globally very few people who are doing this work, which we're trying to change and build capacity because there need to be more people doing this work. But a lot of times people will just contact us and say, I've got this plant, I don't think it can be seed banked, or it's not producing seeds and I need a whole bunch of it for restoration. For those, we'll use tissue culture, which is basically propagating plants in test tubes. It's like water propagation, but instead of water, it's in a sterile test tube with like plant jello. So we can produce a whole lot of plants really quickly that way for people. But yeah, a lot of them are from far away. So we did a long-term project with the University of Hawaii's Lion Arboretum to develop cryopreservation protocols for a lot of their species. Because Hawaii, I mean, plants, we know endangered plants have small populations, but endangered plant populations in Hawaii are on like another plane of existence. Like one of the plants we worked with, there were 3 plants left in the wild, wow, which is crazy.
Wow.
There's only 3 left. But luckily, they've been collecting seeds from that plant for a long time, and we were able to develop cryopreservation protocols. And we have like a lot more genotypes banked than are left in the field, so that in the future they can pull those out and hopefully restore that population whenever it's safe to do so. A couple local species we work with, I mean, there's one that's very local to us, which is the running buffalo clover, Trifolium stoloniferum. So that one we have worked with for quite a while, and it was recently delisted. So it's like a conservation success story where it went from being an endangered species to just being a normal everyday plant. So that one's awesome. We work with a lot of species, Ohio species. We work with a northern wild monkshood. We work with a lakeside daisy. They all have really fun stories. We work with plants from all over.
That's so great. And then tell me a little bit about what's happening with the oaks in your research.
That's been, crazy this week because it's spring, so we're getting all this new oak growth. So all oaks, people don't realize this, but all oaks are exceptional species. We can't seed bank acorns. They're huge. They're very kind of wet. They just don't survive the seed banking process. So we don't really have like a backup bank of oaks, which is kind of crazy because they're such a focal species. And we have so much oak biodiversity here in North America. So we're working to change that by working with a number of gardens and arboreta all across the country. They send us the very new growth. So if you go outside and look at your oak trees right now, depending on where you are, you'll probably see there's this big brown branch, and then at the very tip of it, you'll see just like some new green growth coming out, maybe some like tiny little leaves coming out. We take that as soon as it's growing and we can sterilize it and put it in tissue culture, try to get it growing in tissue culture, and then develop cryopreservation protocols so that we have some sort of backup bank of oaks. Yeah, we're in the throes of that now.
That's awesome. How many species of oak do you think you have?
For this particular project, we're working with 6 endangered or threatened species of oak and 2 widespread, so the bur oak and the red oak that most people know. But how many species of oak we have in our collection right now is probably 20, 25. We have a lot of southwestern species. So when I talk to people about oaks, I'm always surprised because they're like, oh, there's different species of oaks. Like they just think there's one oak tree and like it's just one species. And oaks are crazy diverse. There's so many different types. And out in the southwest, they'll be real shrubby and small, and they almost look like they have holly leaves. Whereas we think of them here as this like mighty, giant, beautiful tree. So it's really interesting to see the full diversity that we work with.
I love that. When I was working in Coral Gables at Fairchild Tropical Botanic Garden, I was like, wait, that's an oak tree? And it looked like a willow leaf. And so that was a really neat lesson about how many different kinds that there are. But I hadn't thought of them as exceptional. Tell us a little bit more about that. What's your definition of an exceptional plant? I know that you mentioned things that couldn't be seed banked. Is that?
Yeah, that's the definition in a nutshell. So if it can't be seed banked using conventional methods, conventional seed banking, you collect your seeds, you dry them down a little bit, and you put them in a normal freezer, like your home freezer. It's minus 20 degrees Celsius. And that works for a lot of plants. Seeds are naturally, evolved to do this, I guess, because they're trying to get the water inside of that seed to be as low as it possibly can. Because water is kind of bad for preserving things long term. Things can go wrong when there's water around, and especially if there's water in something and you put it in a freezer, it freezes and turns into an ice crystal. And so all of our cells are, surrounded by this really kind of delicate membrane, and ice crystals are really pokey and will just poke holes all through it. And then, the cells die when they are defrosted. So if you put a piece of celery in the freezer and freeze it and take it out, it's all wiggly and gross afterwards, because all those nice little delicate cells got punctured by all the ice crystals. So what we're doing with cryopreservation is we're just trying to prevent ice formation any way we can. So for the seeds and the tissues that can't handle the drying aspect of that, instead of physically drying them down, physically removing the water, we can concentrate the water inside of the cells with things that we call cryoprotectants. They're kind of like plant antifreeze. And sometimes, it sounds like a very sciencey thing, and usually we're just trying to put a whole bunch of sugar in there. Because if there's a whole bunch of sugar inside of the cells, then they're less likely to form those ice crystals. We use other chemicals too, but mostly it's sugar. And so we stuff them full of that to kind of decrease the water content or concentrate the water with other solutes. And then on the freezing end of things, we freeze them in liquid nitrogen, so we freeze them really fast and really cold. So instead of minus 20 degrees Celsius, like your home freezer, they freeze in less than half a second to minus 196 degrees Celsius, so much colder. And what happens is they just shoot past that ice crystal formation that would have happened, and the liquids and waters inside of the cell become basically a glass, which is very nice and smooth and keeps all those membranes intact and keeps the whole cell in a state of stasis, basically, until we need to take it back out of cryopreservation again.
Wow. What? Oh my gosh. So you've been doing this for about 10 years. You've probably seen, you've made mistakes.
Oh, yeah.
And you've had successes and probably some failures mixed in there. You mentioned that you need to sterilize the plant material before. How in the world? I mean, you're not getting out the scrub brush and the soap. How do you sterilize a plant?
We almost are. So for a lot of these, what we do is the first thing we'll do is we'll run them under just running tap water. Because number one, that tap water, if it's running pretty fast, is just gonna shoot off some of the contaminants. Okay. So it's kind of gonna work as a physical cleaning. Plus the chlorine in our tap water can also kind of help protect against things like bacteria and fungus. Then we'll usually sterilize them in just a bleach solution. So the bleach that you would pick up from the grocery store, bleach them that way. You can bleach them at various concentrations of bleach and for various lengths of time. Some plants, a minute in bleach is going to be too much. Some plants can handle 20 minutes. It just kind of depends on the plant and the tissue. And then we'll usually treat them with a fungicide and antibacterial and get them into sterile test tubes. But most of this process can be done at home. People can do this in their own home. You have bleach, you can make yourself a little still air box. You could try and do tissue culture at home for sure.
Have you done it at home? I mean, I guess you have access to a lab. Yeah, so you're why would I do that?
I should, but I haven't.
Tell me a little bit more, if we're doing this at home, that we gave them a recipe to make nutrient agar. So that's something I have not tried, and you've made lots of agar.
Yeah.
If you could kind of go through that process, what are we making? How are we making it, and why are we making it?
Yeah, totally. So the nutrient agar is really interesting because we call it plant jello, basically. So it's mostly water that we're giving the plant because plants like water, but we solidify it with that agar or gel in order to give the plant some sort of steady base to hang on to. And then we give them a whole bunch of sugar because, plants are photosynthesizing to produce sugar as their own food. And so why not just give them the sugar so that they don't have to worry as much about photosynthesizing? They're less stressed. They can focus on growing instead. And we give them all the nutrients that we need. So basically like a fertilizer mix, just like put it in there. And then we a lot of times will give them hormones. So plants have hormones just like animals and humans do. They're chemical signals that can travel throughout your body and tell your body to do different things. Plants have them the same way. And we can add those to the media and they'll uptake them into their tissues. And it can tell them, okay, don't make any roots right now. We want you to make a whole bunch of shoots and be really bushy. Or like, now it's time to produce roots because you're going to go back out into the soil. So, and those, a lot of the hormones, not all of them, a lot of them are pretty safe to use, and you can just order them online. So you could also integrate those into your plant tissue culture at home as well.
Whoa.
And a lot of people use them without knowing it already. Like if you're going out and propagating, you'll use rutone or something, that rooting hormone. We use that too. Like that's the same thing that's going into our media. So people don't even realize that they're using plant hormones every day.
That's wild. I love that. Oh my God. Why would someone want to do it at home? Like, because you were saying there's not a lot of people doing it. It would be nice to have more people doing it.
It's a way to quickly propagate a whole lot of plants. You're propagating them clonally. They're basically cuttings, but it's really effective because, again, you're giving the plant everything at once, so it can grow pretty fast. It's anywhere where a leaf is coming out from the stem, there's going to be a little tiny piece of tissue that's like a bud. And so that can grow out and start producing a new stem. That bud contains the stem cells of the plant. We call them meristems. But those stem cells can, they're not differentiated yet. They haven't decided what they're becoming. And so they can become any part of a plant. And so that's what we target to use for propagation, to cryopreserve, because we can induce those stem cells to produce like a whole growing plant later on. So that's what you really want to look for when you're doing home tissue culture, is look for those tiny little buds, usually anywhere where like here's your stem and your leaf's coming out, your bud's going to be right here. Make sure you get that on the media, because some plants will grow from a leaf. A lot of like African violets will. Those are a good one to start with if you want to propagate a bunch of those. But usually you want to make sure that you get a bud. And a lot of people are doing this. A lot of, I think, the home tissue culture is centered around orchid propagation. People love to propagate orchids using tissue culture, and it is a pretty easy way. Orchid seeds are basically just a little floating embryo. They're like dust. They don't have that cotyledon tissue that would feed the growing plant until it can photosynthesize. And so a lot of times orchids are trying to find a fungal symbiont when the seeds land somewhere, so that the fungus can provide the growing plant with any nutrients it needs until it can start photosynthesizing. And then once it's a larger plant, it can start shunting down some of its sugars to the fungus, and they kind of have like a symbiotic relationship. But if you don't have that little fungus, then it's hard to get them to germinate and grow unless you use tissue culture. So a lot of people are using tissue culture to actually germinate these orchid seedlings and get them to grow in habitats that are outside of their natural environment.
Whoa. So you can use a leaf? Because most orchids that I've seen don't really have a clear node. It's like the basal leaves.
Yeah, orchids can be a little complicated to propagate like clonally. But from seed, they're very easy because you can just like throw those seeds on that nutrient medium.
Got you. Okay.
Yeah.
Neat.
They're pretty fun.
My goodness. So exceptional plants. This concept, who made this up?
It was kind of a term coined by our director, Dr. Valerie Pence at Crew. So she's been kind of one of the trailblazers in the conservation of these types of species. I mean, there's people from all over the world that are working on this, but have the whole kind of concept of, a plant being specifically unable to go through conventional seed banking. The term was coined by her, which is really cool. So we're kind of like, people don't know, you're at the epicenter of one type of plant conservation here in Cincinnati, Ohio.
That's amazing.
It is cool. And exceptional plants is such a good term for them, because at the beginning they thought, they're going to be the exception to the rule, right? We think of most plants as being capable of being seed banked, and then there's the exceptions that aren't able to be. And as we've been studying this field for longer and longer and testing more and more plants to see which ones are exceptional, we're starting to find that it's likely that over 50% of plant species are going to be exceptional. So the ones we can seed bank are going to be the exception to the rule, really.
Whoa.
And the scariest ones, like I said, are the ones that seem like you can bank them. Usually if you're doing a banking experiment, you'll bank the seed and then pull it out of the freezer after, who knows, a month, a year, whatever, and it grows and you say, great, these are able to be banked. But if you're not checking them at regular intervals, you might miss that somewhere along the line they've lost viability. And so the fear is that a lot of these seed banks might not have that information and might think that this plant is safe and protected in our seed bank, but it might not actually be viable anymore if it's one of these short-lived seeds. So those are the scary ones that keep me up at night. I feel like, oh my God, we gotta get those.
So when you make a protocol, it sounds like that's part of it, is kind of taking things out. So when you're designing protocols, what does that look like? Do you have a checklist you're working with, or are you you're writing it so that it's not out there yet?
Yeah, so when I'm doing a protocol, I'll usually kind of start with I have a kind of basic cryopreservation protocol where I'll just start and then see how the plant reacts to that, and then kind of adjust things along the way. Maybe it needs more cryoprotectant. Maybe the cryoprotectant is killing it, because the cryoprotectants, they're great for helping the plant survive cryopreservation, liquid nitrogen, but they also can be toxic to the plant. We put things like ethylene glycol in there and DMSO, which can be toxic to plant tissues, although they're great cryoprotectants as well. So there's a lot of just throwing spaghetti at the wall, honestly.
Yeah,
Just trying to figure out what we're doing. And the hard thing about it is that, obviously different species react differently to cryopreservation, but we're finding more and more that different genotypes of the same species react differently, which is so hard, because now not only you can't say, well, we have the cryopreservation protocol for red oak. We're like, well, we have the cryopreservation protocol for this red oak, and we're going to have to do a different one for that red oak, which can be daunting. That's why we need more people to be involved with this kind of research. A really cool thing that has been coming out is the Center for Plant Conservation, which is an amazing organization based out of the San Diego Zoo. They have just launched this free course on applied plant conservation that you can take online. It's one of those MOOC, massive online courses, like Coursera and stuff. And we contributed the whole section on exceptional plants, so you can always learn. And I go into deep detail in there on how to do cryopreservation, and our grad student Max Winklejohn, talked all about how to do tissue culture. So you can really learn the very nitty-gritty of all those techniques on there. They're launching it slowly, so I think they've only released the first couple modules, but I think by the end of the year you should be able to access the entire course. So if people want to learn, more about plant conservation in general and applied, what the people in botanic gardens are doing to do this plant conservation, but specifically cryopreservation and tissue culture, that's exactly where I would send people.
Cool. I'm so glad that people are letting people access this information for free, and that you're well funded, you know, and you've had a secure position to be able to do this work, because it's important. But who knows how long it might take to develop a protocol, and then you've got to get everything out, a few months later, a couple of years later. Our topic today is biotechnology.
Sure.
And so some people might be like, when are we going to talk about biotechnology? Well, we have been, but maybe I didn't outline that right away. So how do you interact with biotechnology?
We tissue culture is considered biotechnology. Cryopreservation is considered biotechnology. I'll give a good little anecdotal story. So we've been involved with work on the American chestnut for a number of years, and so if people are unfamiliar, the American chestnut was one of our real focal species in our North American forests until the early 1900s when this chestnut blight was imported. And it was a little fungus that just attacked every single chestnut. And they all got this blight that basically killed them all. And the plant is now functionally extinct when it was like our forests were covered in American chestnut, 150 years ago. And so there have been over time, there would be like one American chestnut that popped up here and there. And sometimes people would call us and let us know. So we'd run out there, get the pollen from the plant, and then cryopreserve it. So we can cryopreserve pollen too, cryopreserve all kinds of tissues and seeds. And so we had this pollen cryopreserved. We also stored it for experimental purposes in minus 20 degree freezer and a 4 degrees Celsius refrigerator just to see how it responded. And after like 15-20 years of storage, we pulled it back out and found that the cryopreserved pollen was the only pollen that survived the storage at those higher cold temperatures. Did not survive that long-term banking. And so we were actually able to take that pollen that had been cryopreserved, drive it up to the American Chestnut Foundation's farms up in Virginia and use it to pollinate they have some pure American chestnut there, and we used it to pollinate their flowers and produced pure American chestnut seeds using this 20-year-old cryopreserved pollen, which is so cool, right?
So cool. Oh my god.
And so I wanted to go help present this research to the American Chestnut Foundation program, their conference a few years ago, quite a few years ago at this point, maybe like 10 years ago. And I walked in and everybody was kind of side-eyeing me because I was like the biotech person. At the time, there was a lot of people in the American Chestnut Foundation who were like, we're not using any biotech tools because biotech tools are GMOs. There's been a lot of talk about, using GMO technology to put like a gene that was resistant to that chestnut blight into the American chestnut so we can restore it for conservation purposes. I don't do any sort of GMO work. I'm not that well funded. But I think just because I was like the tissue culture cryopreservation person, I was like the biotech person, they were like, no thanks. We don't want anything to do with you. Now, they've been like for so long trying to crossbreed a 99.9% pure American chestnut that has that one chestnut blight resistant gene from the Chinese chestnut that's related, so that we can restore this species. It's been going on for decades, this research. It's so hard to breed trees. It takes forever, right? They live forever. And all of a sudden there were all these GMO techniques, and you know what? Everybody kind of just saw the light and they were like, let's try the GMOs. And they just like, boop, got that little gene in there. There's been a lot of controversy about it recently because, it turns out that they accidentally swapped a label and got the wrong plant growing in all the groves. But still, it's just like another tool we have in our arsenal, although it is kind of like a brave new world of like, what are we doing? De-extincting a plant. Long diatribe on biotechnology.
I love that because I think we're in a space now where it's not one thing.
Yeah.
And these are tools that we can use, and sometimes they're appropriate and sometimes they're not, and that conversation needs to happen, but we shouldn't immediately discount any tool in our toolbox, in my opinion. And so it's not just one thing. So I really like you sharing that, and I'm sorry to walk in there being like, oh. As long as there's good food at a conference.
It was a great conference.
Okay,
they had like swordfish for dinner.
Oh
I was like, I should go to this conference more often.
That's awesome. Good. And I think that what we're interested too is to just kind of keep our ears and eyes open. I think that's really important for everyone. And so if you are interested in biotechnology and want to know more, or just kind of like, still, I'm not quite sure, that's totally fine. I think that the information is important and credible information is important. So finding where you can identify the actual source of maybe funding that went into a certain part of research, or what types of tools that they're using, will help all of us know more about what we're seeing and eating and growing. But I want us to demand that information, because a lot of times I'll read some of this and I don't know what it means. And now we have a resource. We have a network. We're like, hey, what does this mean? Yeah. And if Megan doesn't know, then she can send me to someone else that might. And so Please do continue to be curious and research and ask questions. What's coming up next? What's on the horizon for you?
More oaks. We gotta get through the rest of the spring season. I think once I get my Canadian oaks, that's how I know the season is truly over. It's been a crazy spring because last year we had this nice leisurely, slow spring across the country, and this year it feels like spring hit the entire United States like the last 2 weeks, and it's been wild. So we're getting all those oaks. I think we're up to 500 test tubes of them initiated this year.
Wow.
So we'll see.
Yeah.
Yeah.
And what's your survival rate, do you think?
You know what? Last year our survival rate was so low. I fail so much. I feel like, but it's all data. It's all good to look at. And last year we really focused in on sterilizing our plants and figuring out what are the best methods of sterilization. And we really dialed it in. So we lost like 90% of our plants to fungal contamination, just like mushroom fungus that are in there. But this year, it's looking like, fingers crossed, we're down to maybe around like 30% contamination. So
that's great.
It's slow progress, but it's progress.
That's great. And then the goal for some of these oaks is to do what? Are you reintroducing them to the wild? What's happening?
We are like, we kind of see ourselves as like the cryo preservers. So we are the ones that bank it, and then we just like wait for people to tell us what to do. So sometimes we'll pull them out. Somebody's like, we're ready for them to go back out into the field. We're like, great, we'll propagate them and send them. Or if somebody's like, I just want these propagated right now and not banked, we'll just propagate them and send them back. So we kind of like rely on the land managers who are out in the field to tell us what they want to do with the plants. We're kind of the back end. We'll keep them safe for you until you've figured out, whatever is going on, habitat loss, climate change, etc. in the field.
Wow. I mean, you really are like a bank. Like,
yeah.
It's really, I guess I hadn't thought of like a research lab quite in that way, but it's a nice safe place.
Yeah.
Yeah. Oh my gosh, this has been so much fun. I am going to go ahead and open up the chat. Just a reminder that we will also meet again on May 3rd to talk about ecology as we wrap up our Botany Bistro 1-year anniversary.
I am growing my first GMO plant this year
in
my garden. It's like this new purple tomato that they like inserted a snapdragon gene into.
What?
And it's like a dark purple on the outside, dark purple on the inside, like super anthocyanin tomato. And I'm so excited about it.
Oh,
let's do it. I'll figure it out.
Oh.
I'm so excited.
Can you imagine the tomato sauce that you could make from something like that? Or like
black
pizza sauce? Yeah.
Yeah.
That would be really neat. I love that. All right, Stephanie has a question. Let's see. If I want to start this at home, what seed would you recommend that I start with?
I think the easiest seed to start with is anything that's like a hard seed that you could sterilize pretty easily. Those like tender seeds, like sometimes you go out, if you ever look at like jewelweed seeds, a lot of times they'll be like kind of a more like you could squish it almost. Those are a little harder to sterilize. So anything that has like a hard seed, like a basil seed, tobacco is the classic tissue culture plant. All the tissue culture protocols were developed using tobacco originally. So tobacco is a great one to start with. It's very easy to do in tissue culture. The other thing I would say is if you wanted to start with plant tissue, you could start with an African violet leaf. So just like clip a leaf off of any sort of gesneriad or African violet, sterilize it pretty well because they're fuzzy, so fungus can hide in all those little trichomes on their leaves. But those will just start like popping plants out all over the leaf, which is really cool to see. It just like wants to make a bunch of baby plants.
That's awesome.
Yeah.
Now, do plants get weak over time if you keep cloning them like a copy of a copy of a copy of a copy? Or
yeah.
Do they?
I mean, that's like one of the concerns. We have not seen that as much, but some plants will kind of like start to go a little wonky if they've been in tissue culture or been exposed to hormones for long term. So I try to keep them on pretty low hormones and then dose them out and then bring them back on. Sometimes we will get them restarted, root them, grow them in the greenhouse, restart them again. So Yeah, it is kind of a delicate balance. We have a grad student working on that right now in genetics to see, are there any changes over time or after cryopreservation in these plants? So hopefully not. Pollen is like absolutely one of the things we can go back and find so many things about ancient cultures, like using the pollen that's just like preserved there. It won't always be viable, but it is there that we can like identify what plant or what maybe like genus or family of plant it is by the pollen. We're working on a project right now that's kind of like related to that. That's a little out there, where plants that have been extinct in the wild, but we have herbarium specimens of them. Sometimes people pressed the fruit or the seeds of the plant in the herbarium specimen. So we've been working with Wes Knapp. He has been going and collecting all of these seeds from these herbarium specimens of extinct plants and sending them to us to try and germinate, which is very scary because you have like the last one in existence. But it's a really cool project because it's another way we could resurrect a species that's gone extinct.
That's amazing.
Yeah.
Oh my gosh. Biodiversity is so important because we don't know what we're losing.
Yeah.
And so we really need to keep everything that we can. The pollen kind of woke me up when I started realizing how hard it was, like how long it can stay and how like physically hard it actually is.
Oh yeah. The other species that we're working on a lot of, in addition to oaks, like I said, there's this northern wild monkshood that grows up in Akron, Ohio. That's an endangered species. The people don't even think we have endangered species in Ohio. We do. And that species is one where it's really difficult for us. It's really easy for us to grow it in tissue culture, and then we can grow it in soil and we give it back to Akron, Summit County Metro Parks, and it just like dies. And so we're trying to figure out what's going on, and we think it has to do with the soil. So they live in these like little like cave grottos, and the soil is like sand, and it's many degrees colder than the ambient temperature of the soil is around because it's in this like little wet grotto. And so it's very easy for us to like heat soil up, but cooling soil down is more difficult. We thought, well, maybe that's it. Like it needs that colder soil on its roots than on its leaves, the ambient leaf temperature, to actually make good roots. So our conservation horticulturalist, Mairead Kennedy, built this crazy aquarium that has like basically an air conditioner, like aquarium chiller in it, that like circulates cold water throughout it, all the like sand that's in there, and cools the soil down while like the leaves can stay at normal ambient temperature. And we're seeing that it can actually produce like kind of nicer, healthier roots that way.
Whoa.
But like the lengths you have to go to for some plants, it's so dramatic.
High maintenance plants. I love it. Aw, well, thank you so much, Megan.
Yeah.
I am really excited about continuing our conversation.
For sure.
Learning more. And thank you all so much for joining us today. I had a really good session, and we'll see you in 2 weeks on the Bistro.
Bye.